So one of the things is, so we talked about how to figure out if something is, if an ion is moving like, passively or actively into the cell with an Nernst potential. But also, you can sometimes do some, actual experiments when we're talking about transporters, so things that are not necessarily channels that are moving other molecules. Okay, so you have probably seen a figure like this before. It's actually straight from the cell biology labs, but it is kinetic analysis. So here, in this figure, here you have substrate concentration and then reaction velocity. And sometimes when you increase substrate concentration, measure how much product is produced over time, you can do this kinetic analysis for enzymes and you obtain something similar to this. What are the two parameters that you can measure doing this type of analysis? Parameters, what kinetic analysis? What is this and what is this? Yes. Km and Vmax, right? So, what does Vmax tell you? Yes.
The point at which all the substrate is being used. Or sorry, that substrate occupies all the spots that can be used.
Yes. So Vmax is basically that point in which even if you add more and more and more substrate, the enzyme cannot work any faster because all of the active sites are occupied, right? That's what Vmax is. Even if you add more substrate, things aren't increasing anymore. It's saturated. We're not talking about enzymes. We're talking about transporters. So let's think for a minute about this is glucose. So the glucose transporter is a facilitated fusion transporter that basically binds one glucose at a time, goes, works down the concentration gradient. That's what facilitated fusion is. And so because it's moving one glucose at a time, it's not like a channel, so it opens, grabs a glucose, and then puts it on the inside and does that again. So just to compare it to enzymes, while it's not active site, let's say is that concentration at which, even if you have more glucose, all of the transporters are occupied doing their job. So even if you increase the concentration, they are not working faster. That's as fast as they can. What does Km usually tell you? What does it mean in terms of enzyme? Yes.
It can be an indicator of substrate affinity.
It is an indicator of affinity, right? So basically tells you, let's see if you can remember this. When you have a low Km, like this red graph, versus comparing it to a kinetic analysis that has a higher Km, like the blue line. Does low Km mean higher affinity, or lower affinity? Yes. Higher affinity, right? So low Km values are higher affinity. And higher Km values mean lower affinity. So in general, Km is, this is not an enzyme kinetics class, but it is, half of the substrate concentration, the substrate concentration at half of the Vmax. So basically, how much it takes you to saturate half of the active site. So of course, if you need less substrate to make it work at its max capacity, that's higher affinity than if you need many more of those molecules to make something work at max capacity. So it's affinity. But here's the key. High affinity usually means low capacity. What does that mean? That because the affinity is so high, it very quickly becomes saturated. So you can do your job very well, but also just a slight increase is already saturating the system. It's fully saturated. You're maxed out. Lower affinity usually means, yes, you need more concentration, but also you can work over more time because you need basically the concentration to be that high and the affinity is so low that it's almost never saturated. You can increase, and because you're so slow at doing your job, you can still do it without being saturated. So why is that important for transporters? What we're talking about at the end. So let's assume you do a kinetic analysis, and this is what you get. What do you think about that? What could be happening there? By the way, this is not exactly the reading, so I'm not assuming you know. The correct answer is more like, what do you think? I mean, what kind of that data? Sure. Yes.
So as your like concentration starting to increase, your rate's always going to start to pick up a bit to a higher concentration. It's going to start to level off a bit because it's like the concentration's increased enough for you to start doing it. It hasn't increased to the point where everything is occupied and at max. And then once you like reach that point past the level off, where it just goes straight up and basically reach where you have such a high concentration that everything's going as fast as it can.
So you're saying, okay, concentration increases and you start to max, and clearly something happens here. And what do you think is happening? What do you think is changes? Yes.
It becomes fully saturated.
It becomes saturated, but then what happens here? It's starting to plateau, and then it went, change my mind, I'm not gonna plateau.
It's just the same rate of exchange that it can actually keep up, because it can't keep being shorter.
So do you think the same transporter would be able to change its mind and say, you know what, I'm not done? Because it's let's think you're saturated, right? You're almost working at your max capacity. But then it decides, you know what, I'm not gonna plateau. Instead of plateauing, I'm gonna do it even faster, or not faster. I'm gonna do even more. Yes.
So once you get to a certain concentration outside the cell, I'm assuming that like ion channels open at that point, and as more ion channels open, they're only going to increase forward reaction.
So you're almost there. It's related to concentration. It's almost saturated, and something changes and takes over, and something opens but is not channels. It's two different transporters at least are involved. So you're having a transporter doing its job here at the beginning, and then once this is almost saturated, something else is able to take over and to do the job, to handle higher concentrations. Okay? And that is what happens with sucrose. So whenever you look at the, kinetic analysis of transport for sucrose concentration, you have external sucrose concentration outside of the cell, and then the rate of sucrose uptake. So you have this, and you see how it says the predicted behavior once you start plateauing is this one, but you actually see something like this. So now, based on the rating, is sucrose uptake energy dependent or independent? Let me take this. We're going to talk about this more in detail when we talk about photosynthesis, but this is what happens. Plants uptake CO2. They convert it into glucose. But plants don't really mobilize glucose in the flow, they mobilize sucrose, which is actually two glucoses put together. So that's what the plant exchanges. I am a source leaf. I do photosynthesis, I create my glucose, and then I'm going to mobilize it everywhere in the plant. But instead of moving it as glucose, I'm going to mobilize it as sucrose. So most of the carbon in the plant is mobilized as sucrose. So if sucrose uptake from individual cells, let's say it's in the form and you're a cell, you're uptaking sucrose, is it energy dependent or independent? Dependent. Why do you think is dependent?
Because it relies on other external factors, for example?
Okay, so when it's energy, so you said dependent, right? So it relies on external factors. So yeah, so let's think about transporters. So you're right. So think about transporters. So you're weighing glucose and you said it's energy dependent. You said it's energy dependent. How does when if sucrose enters the cell from the extracellular space into the cytoplasm? Energy dependent. What does that mean? What transports sucrose in an energy-dependent manner? It means that it's against the concentration gradient, right? You're moving sucrose against the concentration gradient. So let's say it's low here and high here. And this is a simple hydrogen. So it's energy dependent again because it's not a pump, but it relies on the gradient, it's secondary active transport. It's as important. Relies on someone else doing the work, putting the excess hydrogen so that it can go down. So yes, it is energy dependent. Can it be energy independent?
Is it energy independent before it starts to plateau and it becomes dependent and it continues and starts plateauing?
Yeah, so you're right. Yeah, so basically that's what happens. At one point is energy dependent, and then when concentration is different, is independent. However, it's slightly the opposite of what you said, but that's the exact correct answer. So what happens is there's a transporter here that is initially energy dependent. Okay. Sucrose initially, let's say, is mobilizing and it's so low outside that the gradient outside versus inside is not that great. So it needs energy to move. So energy dependent. When the sucrose concentration is very small, because it's outside, maybe the gradient isn't great. So you have to move it against the gradient, move it into the cell, so it's energy dependent. However, when the sucrose concentration, if it reaches very high levels, right, it increases, now your gradient changes, and your gradient is now going to be large enough that you can go, you don't need energy, you can go by facility. Because the gradient switched, so now you don't need the C porter, you can actually just do facilitated function. Right? And so basically in this moment, it's not like you can never saturate it, but because the gradient kind of relies, or this transporter relies a lot in gradient and it has low affinity relative to the C porter, you can continue increasing the sucrose concentration and still this will continue. So concentration, movement of sucrose in plants, uptake of sucrose in plants depends on sucrose concentration. At low sucrose concentration, it will move against the gradient, therefore you need a C porter to move it against the gradient. Once the sucrose, and if the sucrose reaches very high levels, then the gradient flips, and now you can move it independently. So in that sense, and that is important, right? If a plant is having a good day, and it's having a lot of sucrose in the phloem, it wants to, like, the cells are going to uptake it. You're not just going to leave that sucrose in the phloem there forever. The plant doesn't know what's going to happen tomorrow. So the individual cells are going to uptake the sucrose. So if it's a good day and there's a lot of sucrose available, then they just have like a backup of, let's uptake these, keep it as a reservoir for whenever we need that sucrose later on to convert it into glucose and do all of it. Okay? So now that we know how this works, let's say that at some point you see sucrose is being moved, but you don't know if it is this or this, this or this. What experiment do you think you could do? What do you think you could do to figure out if sucrose is being moved this way or this way? Oh, I thought you were going to raise your You want to talk about this now, or do you want to talk about it on Wednesday? Yes.
We'd be able to just put it in like a cell during values, so concentrate superstition, see.
Yes, but then again, like let's say for you don't really know. Well, in this case, you kind of know, right? But it's hard for you to know exactly when because you're gonna see sucrose moving in, right? So how do you know? Okay, I put it at 2 millimolar, and then I see sucrose coming in, and I can tell definitely it's passive diffusion or it's simple. Yes.
If you introduce some sort of like non-competitive inhibitor that would inhibit like either the proton pump or the active transport, and then add a high concentration of glucose to see if sucrose, sorry, to see if it would enter the cell or not.
Yeah, so that's one of the things. So when this, it's easier to manipulate this one because it independently relies on ATP and a gradient, right? So think about when we think this gradient as in respiration, right? Electron transport chain, all of those things. We produce ATP and we create a gradient. This at the end is what allows us to produce ATP. So if you can dissipate this gradient, eliminate the gradient, and that stops sucrose moving, then you know it's simple. How can you dissipate the gradient? You can put like a poison on respiration, right? If ATP is not being produced, but you still have sucrose going in, then you're doing facility. Okay? If you dissipate the gradient, that's a bonus. Now let's say by some very intricate method, I don't know if you could do that. You could measure if you're dissipating like a pH gradient because you know you're kind of changing the pH a little bit in that way. So you can basically block ATP production, dissipate this gradient, see if the gradient, the pH is changing relative outside versus inside. So you have different options. Basically, kill ATP production. If everything stops, things were being moved by seaporters. If things continue moving as normal, then things were being moved by the sea. Does that make sense? Is everybody gonna get that on Wednesday? Are you sure? If you're not, I mean, I never do this. This is the time. Let me know now. Because I don't want you to be surprised when you see this on Wednesday. Yeah.
Can you explain that one time?
Yeah. So I'm not gonna go, into. I don't expect you to know intricate details of how this would work, but so this one relies on ATP production, right? Because you need this ATP to create these gradients that you have here. This higher accumulation of hydrogen that then going down the concentration gradient is going to drive movement of sucrose against the gradient. So this is known as secondary active transfer, right? So you need this gradient to be created. So there are different ways. You can say that you can, introduce a poison that blocks respiration. When you block respiration, you block ATP production, right? If you don't have ATP production, eventually so this co-transporter doesn't depend on ATP to transport, but very quickly, if there's no ATP, the gradient is going to dissipate, and very quickly after that, it is going to stop being able to move things against the concentration gradient because you need ATP to create this gradient. Let's say another possibility is, this is more complicated, but you have your plant cells, and then you put them in a solution that is probably high pH, so that there's really not a lot of hydrogen floating around, right? So you don't really have these gradients, a lot of H's, H's, but actually almost no H's, so like high pH. So there's not a lot of this gradient here. So you put your cells in a solution with high pH so that there is no gradient, high hydrogens, low hydrogens. That could also block the transport because you need that favorable gradient of high hydrogens outside versus low hydrogens inside relatively to drive the movement of sucrose against the concentration. That's important. That can be another thing that you could do. Does that make sense? So this one relies on energy indirectly, not directly. This one just energy doesn't bother it. As long as you keep giving it a lot of sucrose outside, it can continue doing the job. As long as the gradient is favorable outside versus inside. Okay? Okay. So let's start talking a little bit about nitrogen, and before I do that, I'll just give you time to review the information. You're gonna okay. So you don't have to do that right now, just yet. Great. Right, so give me a minute. Okay. So we're gonna start talking about nitrogen. Assimilation and nitrogen. So one of the most important things is what I want to make very clear today is that you kind of really fully understand, depending on nitrogen is converted from one to what. Are you consuming energy or are you releasing it in the plant? Because that is we're going to be talking basically about, inorganic nitrogen and nitrate and ammonium and ammonia. And so I want you to have kind of this picture of, okay, she's talking about we're moving this nitrogen into this nitrogen. So is this costing the plant energy or is actually this releasing energy and the plant is actually using it as a source of energy? Okay, so does assimilation of nitrate into ammonium require or release energy? So nitrate is what's most commonly found in the soil. Ammonium is the part that is, or is a component, well actually ammonia, NH3, but ammonium because it's aqueous environment, is actually what's incorporated into amino acids and proteins. So the plant takes it as nitrate and eventually has to convert it into water. Does that require energy or does it release energy? Yeah.
It releases energy.
What?
That's how like the nitrogen is released into the biosphere, right?
Oh, I know about plants. I don't know about nitrogen in the bloodstream. So, is it? I have no idea. Maybe. I mean, maybe. I cannot really contradict you on that because ask me anything or you want, anything you want about plants, but don't ask me about nitrogen in the blood.
No, I said the biosphere.
Oh, in the biosphere. Okay, good. Like, okay. So in the biosphere, is it as ammonium? Is it what you mean?
No, I'm saying like it gets released into the biosphere.
It gets released into the biosphere, but yes, but so does that mean that it releases energy in the process? So it's I wasn't expecting you to know that from right now. So maybe if we can talk about carbon, that is easier to understand. We can then extrapolate that. So when we talk about fossil fuels, we have natural gas, oil, and coal. Comparing amounts, what releases more energy? Natural gas, oil, or coal? Assuming you're starting with the same amount. Yes.
It would be natural gas because oil and coal, while they contain a lot of high energy, but you also have to lose a lot of energy to convert them
Yes.
to get the energy out of.
So natural gas is the more energy efficient. That's why it's cleaner, because basically when I'm getting into our topic about climate change, but it's cleaner in the sense that you can produce the final product is going to be CO2. Always, always, always. So the more energy you can get from natural gas is more energy efficient because at the end you're going to have CO2. But if you only produce a little bit of energy and at the end you have CO2, then you're not very energy efficient because you're releasing a lot of CO2 without being able to extract a lot of energy. So why is natural gas the most energy efficient? This is like concept biology. So the amount of energy released depends on the oxidation states of carbons. The most reduced state of carbon is methane. Carbon full of hydrogens everywhere. The most oxidized state is CO2. You completely oxidized it, it's CO2. The closer to methane you are, we talk about glucose oxidation. We oxidize glucose. Glucose is somewhat reduced, and as we oxidize it, we release energy. What is the final product of glucose oxidation? CO2. Once you have CO2, you have extracted all of the energy from carbon. You cannot extract any more energy from CO2. That's why it's so hard to handle CO2 once it's released, because you want to convert it into something, you have to input energy to make something out of that CO2. Because you have extracted all of the energy that you want. So the more reduced something is, the more energy you can extract because you will extract energy as you oxidize. Okay? Now, how does that relate to nitrogen? Can you order these molecules from the most oxidized to the most reduced? Or the most reduced? If you don't know all the details, don't worry, I had to look it up. But can you at least put something in some order? And we'll fit it in between.
For most oxygens. That was easy. Mhm.
Reverse that.
Once you answer question one, hopefully that's the answer to one.
Making nitrifying bacteria, they're getting energy.
Mhm.
And these ones are.
And then two. Oh, I know two. Okay, that's close. That's right. I have like, I didn't like. I don't know if this is accurate. Can I ask you a question?
Sure.
Okay. So this is the way that I'm gonna.
Yeah.
That is the most positive charge. Like this is.
Yes, so nitrogen, yes.
Okay. So that would be responsible. And then this would be seven.
Yes.
And then zero. But then there's two.
Yes, so basically, it, so yeah, so it's in ammonia, an ammonia. It's it's kind of tricky, but basically, the most hydrogen that you can split, the more you use. So ammonium will be the most.
No.
You also need to answer this. Oh, pretty quickly, we're like, we're done.
It was pretty okay. How your guys has been? We evidently put our leaves back into the first distilled water when we were after the bleach. Yeah, so we decided to just rinse it for a little longer with the other ones. Yeah, so there might be a potential problem with that, but oh well. I'll get you the name exactly of it. I wrote that in my notes when I first got my plants. It was pink chablis. It has a weird Latin name. Like, I googled it recently.
So it's being reduced. Right. Which means that.
Wait,
It's
whatever. Oh, so much fun that scientific names are all in Latin.
Like.
Snake.
Well, if it's
No snake points.
Yeah. I wonder what the same is today.
well, it's just a nitrogen.
Hope he's not sick or something.
That's how I figured out.
Yeah.
Well, I guess that's. When you start talking like that, it's like the numbers. Like to see.
okay, so let's talk about this.
Yeah.
So That's assimilate now. So again, that's assimilation of nitrate into ammonium require energy or release energy. Yes.
It requires energy.
It requires energy, right? So once you put it here, and by the way, ammonia is in here. As we're going to see, basically, many times when you have ammonia in an aqueous environment which cells are 98% water, you're going to actually have ammonium. That's why we have this. So again, anytime you're going from here to here, because you're reusing something, you require energy. Anytime you're going from here to the more oxidized state, you are releasing. So basically what happens is you have, when we're talking about nutrient assimilation plants, and we're not going to talk about sulfur, just nitrogen. When you're trying to convert these inorganic molecules into organic molecules, call it amino acids, proteins, all of these compounds, then you are basically consuming energy in that process, right? You're making more complex molecules. And also, one thing that the book mentions is that so much energy, for example, is consumed in this process, from fully oxidizing, from fully reducing some of these agents like N2 to NH4, that if you have the opposite reaction, it can be very, very expensive, right? Some of these reactions, if they are in control, because in a cell, this happens step by step. If you did like industrial, you have to control it because so much energy is released in the opposite direction that they can be explosive. That's why, I don't know if you watch this in the news, I don't even know what I'm talking about it, but there was recently, like a year or 2 years ago, a terrible explosion in Lebanon, in Beirut, that it was terrible. And it was these, where storage places where fertilizer was abandoned there. Fertilizer has a lot of ammonium nitrate. And then that was basically what happened here, was all of these abandoned fertilizer. So you have that combustion process and it's explosive. Okay, so this is the nitrogen cycle. And because plants, yes, carbon, and we're going to talk about photosynthesis, the nitrogen is the next limiting factor. So the nitrogen cycle is very important for plants. So where do you find the largest reservoir of nitrogen? Yes.
The atmosphere.
In the atmosphere, right? So it's in the atmosphere. So when we look at the components of air, so basically it's lots of nitrogen present here. So that's why nitrogen fixation is one of the most important processes that happens here. So you have nitrogen basically produced as a gas right in the atmosphere. And then you're going to reduce it to ammonia or ammonium in this case. That is what's the component of chlorine. So how can that atmospheric nitrogen become available to living organisms? How do you grab that N2 and make it NH4? Yes.
It's usually converted by bacteria.
Yeah, so we have bacteria, right? So we have, this is like the industrial process. Clearly, if you are going to do this industrially to produce something like fertilizers and things like that, then a lot of high temperature, high pressure, you need a lot of energy put in to make this happen. Or you can have the biological nitrogen fixation. Don't worry about this. We're not going to look at the equation any of these things. But you can see you have, from atmospheric nitrogen, then you have a lot of energy, and then eventually you have your ammonia. That when you have ammonia in the aqueous environment, you usually have ammonia. And that's what you usually talk about. So nitrogen fixing organisms convert atmospheric nitrogen into ammonia. Does this release or consume energy? Yes.
Consume.
It consumes energy. right? We're trying to go from here to here. You have to consume it. So you know, whatever. So a lot of bacteria or the nitrogen put in the soil. So now you have nitrogen in the soil, and then it is converted to ammonium in different ways. So, for example, plant and animal wastes, right? So once you have decomposing organisms or organic matter, then that will release some of the ammonium that is now available in the soil to take, and that process is known as ammonification. Then once you have ammonium, you can have some of that ammonium immobilized. But immobilized means because the plants have it in roots or soil microbes, or it is immobilized because it's attached to other particles in the soil that just basically make it not accessible. And then you can also have some nitrifying bacteria that take some of that ammonium away from the plants. Right? They are using it and the plants can use it. And so they convert ammonium to nitrate. Now, let me see what my question is. Nitrifying bacteria convert ammonia into nitrogen. Is this nitrogen a source of energy or an electromagnetic? So if you're going from ammonium to nitrogen, yes.
So it'd be using it as a source of energy.
Yes, so a source of energy. So now every time you hear of nitrifying bacteria or bacteria that can use ammonium, right? So they are actually using as a source of energy. You don't necessarily need glucose, it's not that they don't have to, but they can use other sources to oxidize them and produce energy at the end. So you're releasing energy. And then another, kind of completing the cycle, then what we have is the nitrifying bacteria have nitrate, and then basically they release it back as atmospheric nitrogen in the middle of the cycle. And so we have the process of nitrification. So basically now, the nitrifying bacteria, if they convert nitrate to atmospheric nitrogen, what are they doing to this nitrate? Are they reducing it or oxidizing it? They're reducing it. So now next time you hear the nitrifying bacteria, what happens is in an anaerobic environment, instead of using oxygen, they will use nitrogen, right, as their electron acceptor, and they are reducing their NO3 to atmospheric nitrogen. It reduces it, it accepts their electrons while they do, their anaerobic respiration, and then it releases atmospheric, into what happens. So, you reduce. So let me put this into perspective. There is ammonium in the soil because the organic matter, there's a process of ammonification. And we talked a couple of days ago, I don't expect you to remember, that fertilizers contain two different sources of nitrogen, ammonium and nitrogen. Now, if plants uptake nitrate, which they do actually, is their favorite source of nitrogen. Why do they don't just uptake ammonium? Why? If they have to spend energy, uptake nitrogen, then spend energy reducing it to ammonium to then incorporate it into their amino acids. Why don't they just prefer and only uptake ammonium? Yes.
It's toxic. It builds up enough. It's toxic.
Why is it toxic?
I think if the buildup is high enough, right, it becomes reactive, and then it starts to actually do the opposite, right, and break down instead of helping the plant build. That's a very vague answer, I know. But
Yeah. So if it accumulates, it starts doing toxic things. Yeah, that's correct. It's bad. So it's not that the plant doesn't like ammonium. It clearly produces ammonium and then incorporates it. But why doesn't it just accumulate ammonium if it's easier for the plant? It's toxic. Why is it toxic? Yeah.
Because the sugars have to move down from the leaves and the roots before the ammonium can be metabolized.
So the ammonium actually moves from the roots up, right? Because ammonium is picked up from the roots, from the soil. So you're moving it up. So if the soil has ammonium and fertilizers were clearly manipulating, we're creating fertilizers, why don't we just feed them ammonium if it's easier for the plant? It has to waste energy because you pick up nitrate, you have to do all of this, and then this can be incorporated into amino acids. Why doesn't the plant just uptake ammonia?
It would release too many hydrogens, which would make it an acidic environment or a high electron environment, which would make it poisonous.
Yeah, so it's very toxic because it dissipates the gradient. So this is what happens. We have been talking about this gradient for a while. Okay, so we have this cytoplasm here, this is the cellular space here. And we said that plants have, first of all, they have um, the H type ATPase that pumps hydrogens outside. Right? And so these higher content of hydrogens outside, remember we talked about how is the pH on the extracellular space relative to the cytoplasm. I said the extracellular space had a lower pH than the cytoplasm because these. So we a few minutes ago talked about some of these hydrogen is important for seaport transport of sucrose. Also glucose and many things rely on this gradient to move into the plant. You need this gradient here to move things into the plant and to take toxic things out of the plant. If you don't have this gradient, then the plant cannot import many things because many import of many components into the plant relies on this gradient to move things down the concentration gradient or by co-transport. So what happens with ammonium is this is lumen space macro cytoplasm. Oh, okay. So this is basically what's happening in the cytoplasm, right? If you have a lot of ammonium accumulated in your cytoplasm, right? What's happening here? Ammonium, right? You have excess of ammonium, and what you have is because the cytoplasm has a different pH relative to the outside, what happens is this ammonium can actually dissipate that pH that you have there. Why? It combines, with your hydroxyls again because the cytoplasm is relatively higher pH than the outside, right? So by dissipating this is stealing some of these OHs. So this gradient of higher pH versus lower pH is being dissipated. Stealing some of the OHs here. Ammonia can easily diffuse, steal some of these hydrogens, and be converted into ammonium. So this gradient of higher hydroxyls inside of the cell versus higher hydrogens outside of the cell, it dissipates this gradient that you can see here. And then if you dissipate that gradient, because ammonium is basically stealing some of these, again, OHs versus H's that are creating the gradient. You dissipate the gradient, and then you impair the plant from importing many of the things that rely on this gradient to be moved into the cell, or again, toxic things that are meant to be pushed outside of the cell. You need this gradient of higher H's. So there's relatively higher pH in the cellular space, and also higher pH on the lactate relative to the cytoplasm. So by associating with some of these hydroxyls and dissipating the gradient inside and outside, that's what it does. Okay. So now we said, okay, so plants uptake They cannot fully accumulate it, so they uptake nitrate mostly from the soil. They kind of take some ammonium, but not too much. So they uptake nitrate, and that's what's known as nitrate assimilation. So you uptake nitrate from the soil, and then you're going to reduce it to ammonium to, again, incorporate it as part of all of the other, basically, organic compounds in the plant. So it's divided into parts. First, nitrate is converted into ammonium. And then we're going to talk about this later on Monday. Ammonium is assimilated into amino acids. So which enzymes are involved in nitrate assimilation? And in which organelles is their activity more important? How do you go from here to here? There are two enzymes. One does this, the other one does this.
Yes, but with the ammonium assimilation and lysosomes.
Oh, yes, but we're focusing on this. We're gonna talk about this in Monday. So, yes, so let's focus on this part. How do you go from nitrate to ammonium?
So is one of the enzymes or reductase?
Yeah, so both of them are reductase. Right? You're reducing from here to here and here. So one of them is nitrate reductase, and you find it mostly in the cytoplasm. I always have problems trying to pronunciate nitrate and how do you pronounce NO2? I know how to write it.
Is it nitrite?
So how is nitrite different from nitrate? I mean, I hope you can tell the difference. That's why I will always write it. So nitrate reductase reduces nitrate to nitrite for this. But this is very toxic. NO2 can be toxic for plants. So they very quickly want to convert this into this. So nitrate reductase does it in the cytoplasm. And then this NO2 is transported into the chloroplast quickly, kind of isolated. And then nitrate reductase converts NO2 to ammonium in the chloroplast. Okay? So make sure we're gonna leave this here. But then we're gonna start talking about this on one. And we're going to see carbon and nitrogen assimilation and the specific process, one thing I can tell you about Monday is this process, this specific process, how this gets converted into this, that's what we're learning about. Okay? So please leave your worksheets here and have a great weekend and I'll see you again.
It's so bad.